Hello everyone, welcome back to Pharmaceuticalmanufacturing. Today's topic is dirty hold time and clean hold time studies. Why do we do them? How long should they be? And what happens if we ignore them? Let's break it down in a simple and practical way. What is hold time in general? In pharmaceutical manufacturing, every minute counts, but sometimes equipment can't be cleaned immediately after use, or sometimes it stays clean for some time before the next batch starts. This gap in time is called hold time, and depending on whether the equipment is dirty or clean, the requirements change. What is dirty hold time study? DHT. Dirty hold time is the period between the end of manufacturing and the start of cleaning. While the equipment is still dirty, this period is critical because residue and moisture create ideal conditions for microbial growth. Why DHT is required? Key reasons: residue hardening, microbial growth animation, cross-contamination symbols. Dirty hold time studies are required for four major reasons: to prevent residue hardening. If equipment sits dirty for too long, product residues can dry, harden, and become nearly impossible to clean later. To control microbial growth, wet and nutrient-rich surfaces are like a buffet for microorganisms. To avoid cross-contamination, any residue left behind can enter the next batch, a huge GMP violation. To define the maximum allowed time before cleaning. This helps create a controlled and validated cleaning schedule. Even after being dirty for some time, the equipment must still be cleanable effectively. Typical dirty hold time duration. Typical dirty hold time limits in the industry are 24 hours, the most common, 48 hours, 72 hours, and some facilities go up to 5 to 7 days, but only if strong validation supports it. Because dirty surfaces carry high risk, DHT durations are usually short. For example, if manufacturing ends at 6 p.m., and your validated DHT is 24 hours, your cleaning must start before 6 p.m. the next day, not a minute later. This keeps equipment safe, cleanable, and GMP compliant. What is clean hold time study? CHT. Clean hold time is the period between the completion of cleaning and the start of the next batch. Here, the question is, after cleaning, how long can equipment remain clean and safe? Why CHT is required? Key reasons: dust prevention, equipment covering, microbial testing charts. Clean hold time studies ensure that the equipment stays clean over time, without new contamination. There is no microbial growth during storage. Closed or covered equipment remains residue-free. GMP rules for equipment cleanliness are met before starting the next batch. It confirms equipment stays safe and production ready. Typical clean hold time duration. In most pharma facilities, CHT limits are 72 hours, 3 days, 7 days. Most common, 14 days. In some advanced facilities, for dry and closed systems, up to 30 days, since clean and dry surfaces carry lower risk. CHT durations are longer than dirty hold time. Let's say your validated CHT is 7 days, if the equipment was cleaned on Monday, you can use it safely until the following Monday, as long as it stays closed and protected. What happens if hold times are not validated? If a facility does not validate its hold times, it can lead to microbial contamination, hard to clean residues, batch rejection, regulatory observations from USFDA, EMA, MHRA, severe compliance issues. So both DHT and CHT studies are essential for GMP compliance, patient safety, and product quality. Thank you for watching. I hope this video helped you understand the importance of dirty and clean hold time studies in the pharmaceutical industry. If you found this useful, don't forget to like, subscribe, and share it with others in the